very happy good evening. So this is Krishnaveni and we've been talking about the story of life on earth, and that's biology. So welcome to my session. So today we are starting a new topic. So the new topic is cell, the unit of life. So it's been a pretty long time since I taught this chapter. So when I started my teaching in this YouTube channel, I started off with this chapter. So after that, I never had an opportunity to talk about this chapter. It was revision or it was directly Menti quiz, but the theory part I have never taught again. So once again, I have got an opportunity to teach this chapter. So cell, the unit of life, the most simplest and the most important topic that you should start with in biology, because it talks about the importance of a cell and how superbly the cell functions. Okay, so today we are starting unit 3 under your syllabus wrap-up series. So 2 units are done. So we are starting with the 3rd unit. Yes, hi Anthony Abhi, a very happy good evening. So thank you so much for joining my lecture today. So please do like, share and subscribe. So cell, the unit of life. So lecture 1, so 2 units are done. For 1 unit I have already conducted the Menti. The 2nd unit is done, that is morphology, anatomy and your animal tissues, followed by a cockroach. Yes, so this was completed in unit 2. So tomorrow at 6:30 p.m. you have a mega Menti quiz for your unit 2 of your syllabus wrap-up series. So please join me for the same tomorrow at 6:30. So the winner will receive access to free mock test series. Okay, eagerly waiting. Alright, Anthony Abhi. So this is lecture 1. So let's go forward. So this is my schedule for tomorrow. So we started off with the 5 steps of glycolysis in the previous lecture, that is yesterday. So tomorrow we'll finish off glycolysis, move forward with fermentation. So talk about what is link reaction. So this is at 4 p.m. So please do join me tomorrow as well at 4 p.m. And tomorrow at 6:30 we are having a mega Menti. That is for your syllabus wrap-up series. So your portion is known. So it is morphology, anatomy, animal tissues and cockroach. The one who wins this Menti will get access to a free mock test series. Hi Nishrag, yes, a very happy good evening. So thank you for joining my session. So please do like, share and subscribe. I hope you are doing well, Nishrag. Yes, hello, hi. Fine, so this is my schedule for tomorrow. So please prepare these 3 chapters. So I have given enough questions. I have taught this time and again. So please go through. So tomorrow almost around 20 to 40 questions you will have. So the winner will get access to a free mock test series. So prepare seriously. Alright? Yes? Okay, so shall we move forward with cell, the unit of life? Yes, cell, the unit of life is a huge chapter compared to your cell cycle or your biomolecules. It is pretty huge. So we will also go slowly because after a long time I am teaching this. So I don't know if you remember. So we are not hurrying up. So I will take 3 lectures to finish this chapter. So today the first lecture will be very simple. So I am not going deep. Alright. So shall we go into cell, the unit of life? So how many of you still find this cell, the unit of life, difficult? Ma'am, this chapter is confusing. I am fine with plant physiology, I am fine with animal physiology, but this unit is what is tough. So under this unit, so you have 3 chapters, that is cell, the unit of life, cell cycle, cell division and biomolecules. How many of you feel this chapter is tough? Yes, you will get access to a new mock test series. Okay, Nishra? Yes, so we will not give access to the same mock test series, but a different one. Yes, Dharani? Yes, a very happy good evening. So thank you so much for joining my lecture. So please do like, share and subscribe. So shall we go forward with cell, the unit of life? So how many of you still feel this chapter is tough for me? Yes, anyone out here who says, ma'am, yes, this chapter is tough for me? Yes, it's okay. Because you have to remember a lot of things here. So Anthony Abhi says, no ma'am, EC only. Okay. Fine. So shall we move forward? So here we go. So we shall elaborate on. So what are the topics that we will talk about in this chapter? So what is a cell? So why should we study about a cell? So what are the features of a cell? So how is a cell useful in our everyday life? So without cell, can we work? If that's not possible, so we are going to define what is a cell and what are the constituents or what is the architecture of a cell. Okay, ma'am, I think in this, okay, hi Shimpi, hi. How are you? I am good, thank you. You didn't come for my previous lecture. I hope you're fine. Did you sleep off? So anyways, thank you you so much for joining my session. So please do like, share and subscribe. I think in this lesson out of NCERT are coming like what? What questions did you come across outside NCERT? Is it your gram-positive and your gram-negative bacteria? So Anthony B says, if I teach anything, everything will become easy. Alright, okay, so thank you for having that faith and trust in me. I will never disappoint you, okay? Yes, so we will start off with talking what is a cell, then we'll move forward and talk about your cell theory. So who proposed the cell theory? What are the postulates of the cell theory? Then overview of the cell. So who are the members inside a cell? So if you look into a palace, so who are the members inside a palace? So it is something similar to that. So then prokaryotic cell. So how is a prokaryotic cell different from a eukaryotic cell? So prokaryotic cells are pretty less developed compared to a eukaryotic cell. So these are the topics we will talk about. So today we will talk about till here, at least we will begin with your prokaryotic cell. Your eukaryotic cells are pretty long, right? Microbodies coming out of NCERT, yes, you have some terms like your glyoxysomes, peroxisomes, spirosomes. So those are microbodies. Yes, those come. So we will discuss all that, so don't worry. Yes. So there is net problem, so you couldn't come. It's okay, you don't have to be sorry. So Pooja and me were missing you, so that is the reason I asked about it. Okay? Yes. Yes, Nishrag, I will teach you about microbodies, so don't worry. Okay? So here we move forward. So what is this? So what is that makes an organism living? So going back to your first chapter in class 11, that is your living world. So what makes an organism living? So how can you feel that you're living? How can you feel? Yes, of course, all of us are breathing. Yes, so we have a diaphragm moving up and down. If a cow is sleeping, you can feel the up and down movement of the diaphragm. If a dog is sleeping, yes, you can feel the up and down movement of the diaphragm. But what is that makes an organism living? So what makes you feel that yes, you are living, you're lively? Yes? Yes, good evening. So thank you for joining my session. Yes, answer this. What is that makes an organism living? Hi PUBG Gamer. Yes, after a long time, I hope you are good. So thank you so much for joining my section. So please do like, share and subscribe. Hi Antonia B. So metabolism is what makes an organism living. But where does this metabolism happen? Yes, where does this metabolism happen? Yes, so it is the cell that makes us feel living because cell function performs all the necessary function. Okay? I don't very much about this, ma'am. My seniors told that some questions come out of NCERT. Okay, like Nishrek said, it is your microbodies. We will discuss about your microbodies. Okay? Okay, so thank you, Shimpi. Yes. Communication and metabolism. Okay, so what makes the metabolism possible? That is our cells, because metabolism can happen inside our cells, right? So that is the main reason. All right, so yes, the answer is cell. So this is what makes us feel lively. So this is the basic unit of life. So they often say no. So when you have the science memes, especially your biology memes, so whenever you're feeling lonely in the world, so remember you have 10 power 14 cells in your body which are trying to save you, which are trying to protect you. So you're not alone always, right? So that is so true. So your body has this healing power. So every cell in your body is trying to keep your functions normal. Every cell in your body is fighting to make you fit and healthy. But what are we doing on the other hand? Spoiling our body. How? Drinking, smoking, having unhealthy food, all that. So we are putting ourselves in a lot of danger and a lot of struggle. So let's see how beautifully your cell functions. So moving forward, so your cell can be unicellular or multicellular, right? So unicellular means one cell makes up an organism, only one type of cell. So multicellular, you have many types of cells which make up an organism. For example, we humans are multicellular organisms, right? So we have so many types of cells, like your nephrons, your RBCs, so you have your hepatic cells, you have your cardiac cells, you have your intestinal cells. Yes, all of them coordinate together to give you a perfect function, right? So this is multicellular. So moving further, So what is cell theory and who gave the cell theory and what are the postulates of the cell theory? So for example, when you talk about a particular concept, for example, previously we were talking about chemi-osmotic hypothesis, right? So chemi-osmotic hypothesis states that you have higher H plus concentration inside the lumen. How you have more H plus concentration? You have three ways. So similarly, features of a particular theory is known as your postulate. Okay, so here let's see. So Anton von Leeuwenhoek was the first one who saw and described a live cell, because he was the one who discovered a microscope. Okay, he was the one who first saw and described a live cell. So later Robert Brown came and he discovered the nucleus that is present inside the live cells. So then you have Matthias Kleeden. So he is another scientist. So he was a botanist, to be sure. To be very specific, so he worked on plant cells and he found out that plants are composed of different kinds of cells which form the tissues of the plant, right? You have meristematic cells, you have permanent cells, you have your simple tissues, you have your complex tissues. So that is what Matthias Kleeden found out. So then you had another scientist, that is your Theodor Schwann. So Schwann was a zoologist, so he worked on animal cells. So different types of animal cells, they have a thin outer layer known as plasma membrane. So how it all started? It all started with the discovery of microscope. So who discovered the microscope? Anton von Leeuwenhoek. So he discovered the microscope. So he first saw and described a live cell. So then someone came and discovered the nucleus, and that scientist is your Robert Brown. Okay, so then you have Matthias Kleeden. So he was a botanist, so he worked on plant cells and he said that all plant cells are composed of different kinds of cells which form tissues of the plant. Then Theodor Schwann, so different types of animal cells and they have a thin outer layer known as plasma membrane. So these two scientists, that is your Kleeden and Schwann, we say commonly. So Kleeden and Schwann, they formulated what is called as the cell theory. So they did not explain how cells are formed, how a new cell is formed, right? So that was the mistake that they made. So later a third scientist came, he is called as Rudolf Virchow. He said that cells divided and new cells are formed from pre-existing cells. So when I say I have a mother cell is dividing to give two new daughter cells. So the new cells come from the pre-existing cells, right? So this is what Rudolf Virchow proved. So your cell theory was initially formulated by Kleeden and Schwann, but since they failed to tell us how the cells are formed, so you have the involvement of a third scientist who is called as Rudolf Virchow. He said that cells are formed from pre-existing cells. Thus, the modified postulates of cell theory as defined by Rudolf Virchow are: all organisms are composed of cells and products of cells. Right? So all organisms, so a unicellular organism has one cell, so we multicellular organism are products of cells. Everywhere we have cells. So this is my palm, so if I swipe like this, yes, I am losing a lot of cells. I have cells on the surface. So if I rub my cheeks, yes, I am having cells. So I am clearing off a lot of cells right now. So all organisms are composed of cells and products of cells. Okay, so this is the first postulate. Number 2, all cells arise from pre-existing cells. Yes, for me it's very hard to remember the scientist's name. Okay, so you don't have to remember the first two scientists, that is Anton von Leeuwenhoek and Robert Brown, but you should remember Kleeden and Schwann. Both are SS, right? So remember, this is botany. So botany is a long term. So it is Kleeden. So zoo is a small term, so Schwann. So Kleeden and Schwann gave the cell theory, but they forgot to tell how the cells, or they failed to express how the cells are formed. So you have a third scientist who is called as Rudolf Virchow. So this Rudolf Virchow, he said that cells are formed from pre-existing cells, and he gave two new postulates, that is all living organisms are composed of cells and products of cells, and all organisms arise from pre-existing cells. So these 2 points you should remember. Clear? So shall I move forward? Yes? Okay, so this is about your cell theory, right? So so here mostly they won't ask about your Schleiden and Schwann, but they ask about your Rudolf Virchow. So who was the one who said that cells are formed from pre-existing cell? So please remember Rudolf Virchow. So the two postulates of cell theory that you read now were given by your Rudolf Virchow. Okay? So moving forward to the next one, overview of the cell, the prokaryotic and the eukaryotic. Okay, so we are not naive to biology. We are not new to biology. So we have read complex chapters, we have read complex terms. So now everyone will give me one point of the difference between prokaryotic cell and your eukaryotic cell. Quick! So this is something very basic that you would have studied in class 8th or 9th. Yes, point number 1. So what is the difference between a prokaryotic and a eukaryotic cell? Make it quick. Yes. What is the difference between a prokaryotic and a eukaryotic cell? Quick. So Shimpi, give me 1 point. Antonia B, give me another point. So Dharani, give me a third point. Yes. Shimpi, what is the first difference between a prokaryotic and a eukaryotic cell? Yes, come on. We are not new. Which is unicellular, which is multicellular? So all prokaryotes are unicellular. So your kingdom Munira, so they can be unicellular or multicellular because you have your kingdom Protista. Yes, then next point. No nucleus. Which one has no nucleus? Eukaryotes are only multicellular, there are no unicellular eukaryotes. Example, yeast. Yeast is a unicellular eukaryote. So what about your protozoans? Sorry, protistans. So Shimpi, prokaryotes have no nucleus, bacteria has no nucleus. So if a cell has no nucleus, then it is enucleated like your RBC. So your prokaryotes have no nucleus. So which means replication, transcription, translation, that is central dogma of life, does not happen in your prokaryotes. Yes? What is the second difference? Yes, make it quick. Shimpi says no nucleus. Oh my god, I didn't know. They do not have well developed nuclear membrane. So nucleus is present but nuclear membrane is absent. So here they have well developed nuclear membrane. So this is a cell. If this is your nucleus, it contains your chromosome and your DNA. So there is a membrane surrounding your nucleus in case of your eukaryotes. Your prokaryotes do not have this membrane. So that is the difference. Yes, apart from this, what are the difference you have? Give me one more. One important difference between prokaryotes and eukaryotes. I guess still people are not confused with what are genes, what is a DNA and what is a chromosome. So these are clear. So these are some of the basic things. So which is inside what? So where are chromosomes, where are DNA, where are genes? Can anyone tell me the correct correlation? Yes, make it quick. Cell wall. So prokaryotes have a cell wall. So that is the difference between a plant cell and an animal cell. So plant cell and animal cell both are eukaryotic cell. Your plant cells have a cell wall. Your animals do not have a cell wall. Okay, so that is not a difference between prokaryotic and your eukaryotic. So composition is peptidoglycan. Okay, I agree, bacteria has a peptidoglycan layer, that too only gram-positive bacteria. Yes, one most important point in your prokaryotes and eukaryotes, and please tell me the correlation between these as well. Quick, make it quick. What is the most important difference between prokaryotes and eukaryotes? Yes, there is no difference. They have no membrane bound organelles. So hi Satyendra, yes, thank you so much for joining my session. So please do like, share and subscribe. Which one is big? Of course your eukaryotes are bigger compared to your prokaryotes. So eukaryotes have well developed membrane bound organelles. So organelles, what I mean? Yes, class is not at 6:30, it starts at 6. So every day on alternate days, that is MWF, classes from 6 to 7. TTS, when we are having a menti, it is at 6:30. Okay, Satyendra? Yes. So what are organelles? So this includes your mitochondria, your chloroplast, your endoplasmic reticulum. So all of these are membrane bound organelles. Okay, so that is absent in case of your eukaryotes, but it is present in case of your it is absent in case of your prokaryotes and it is present in case of your eukaryotes. Done? Okay, so quickly tell me the correlation between this genes, DNA and chromosome. So which is present inside what? Quick, make it quick. So where are genes present, where are DNA present and where are your chromosomes present? Yes, I know we have 23 pairs of chromosomes, but where is it present? If I want to isolate your chromosomes, how would I do? If I want to isolate your gene, how would I do? Come on, make it quick. Quick. What is the difference between these three things? Genes, DNA and chromosome? Yes? No idea? It is high time you should know it well, because we have completed genetics so many times. Okay, what is present inside the nucleus? Chromosome, DNA or gene, which is present inside the nucleus? Quick! Why so slow? Genes contain chromosome and DNA. Oh my god, no, it is vice versa. So your DNA is the genetic material, right? So any segment on your DNA that codes for a sensible protein, this sequence is known as your gene. So genes are present on your DNA. So DNA is too long. So the length of your DNA is 2.2 meters, but the diameter of your cell is 10 power 6. So it is in micrometer range. So your DNA are packed into your chromosomes and they are present inside your nucleus. Okay? So gene is a sequence on the DNA that codes for a protein. So DNA is your genetic material which is too long. So it is compacted inside the cell in the form of your chromosomes. Okay? So chromosomes are the condensed form, chromatin are the non-condensed form. So this is the difference between these terms. Clear? Yes? Yes? No confusion? So will you remember this? Write down. So gene is any segment on the DNA that codes for a protein. So DNA is deoxyribonucleic acid. So that is your genetic material. Your DNA is too long. So it is compacted inside the cell in the form of chromosomes. So chromosomes are the condensed versions and chromatin are the non-condensed form of your chromosomes. Okay? Yes? Okay, so overview of the cell, the prokaryotic and the eukaryotic cell. So this is your eukaryotic cell and that is a prokaryotic cell. So here we go. So both of them have nucleus, but the nucleus is not well developed in case of your prokaryotes, but it is well developed and it has a nuclear membrane in case of your eukaryotes. Okay. So in both you have a semi-fluid matrix called as cytoplasm. So if you remember, recently we were talking about photosynthesis. I told you about chloroplast, right? So chloroplast is a double wall structure and the space inside the chloroplast is called as stroma. So this space inside the cell membrane, okay, so that is known as cytoplasm. The semi-fluid matrix is your cytoplasm. So what is your plasma membrane? So plasma membrane acts as a boundary between the thus separating your cytoplasm and the nucleus from the cell wall. So pay attention. Okay, I don't have a blank slide. So please make a note of these things. Yes. So what is the plasma membrane? So what is a cytoplasm? So you have a cell. So this space inside the cell is your cytoplasm and your cell also has a nucleus, right? And the cell also has a cell wall. So plasma membrane separates the cytoplasm and the nucleus from your cell wall. Okay, so cytoplasm is the semi-fluid matrix present inside the cell. So this is plasma membrane, this is cytoplasm. You can say plasma membrane demarcates your cell wall and the cytoplasm and the nucleus of the cell. Okay, yes. So eukaryotes have membrane bound distinct structures called organelles like your chloroplast, like your mitochondria, whereas your prokaryotes have no distinct membrane bound organelles. So you have mitochondria, you have your endoplasmic reticulum. So here there's nothing, only a nucleoid is present. So why it is called as a nucleoid? Because it is bit scattered and it is present only with your histone proteins. So prokaryotic cells, they lack membrane bound organelles. So ribosomes are non-membrane bound organelles that are present in both prokaryotes and eukaryotes. Just like prokaryotes and eukaryotes have distinct differences, they have some similarities as well. So both your prokaryote and your eukaryote, they have non-membrane bound organelle that is known as your ribosome. So all living cells have ribosomes because ribosomes are involved in the translation of proteins. So what is the example that I will explain? So if I have a fan, so what is the ultimate purpose of a fan? To give me good air. When I'm sweating, I turn on the fan so that I don't sweat anymore, right? So what is the agenda of having a having a fridge? So that I can store my items so that it does not get spoiled very quick, right? So similarly, the entire purpose of your cell is to produce proteins. Yes, your cell undergoes a lot of metabolic process, but at the end of the day, the main function of your cell is to produce proteins, and for that it has to undergo a process known as translation, and for translation the major players are your ribosomes. So without ribosomes, your proteins will not be produced. Thus all living cells have ribosomes with them. Clear? Yes. So animal cells contain another non-membrane bound organelle that is known as centrosome. So centrosome consists of two cylindrical structures, that is your centriole. So your prokaryote and your eukaryote, so both of them have non-membrane bound, which means ribosome is not surrounded by any membrane. So they have non-membrane bound ribosomes. Okay? Yes. Apart from that, your eukaryotic cell has another non-membrane bound organelle that is known as your centrosome. So what is your centrosome? So centrosome has two cylindrical shaped structures which are known as centrioles. So the function of these centrioles are to pull the chromosome apart during the anaphase. Okay, yes, so that is the function of your centrosome. Your ribosome is involved in your protein translation. Clear? Yes, so you now know what is ribosome and what is centrosome. So don't confuse centrosome. So this is your centrosome. You have another term known as centromere. A centromere is different from a centrosome. So centromere is this portion where the arms of the chromosome are attached. This is your centromere and this is your centrosome. Is the difference clear? Yes, everyone? So can I move forward to the next point? Yes, no doubts? So these are some of the basics which I want you to remember and never forget. Okay? Yes, centrioles are right angles to each other. So different shapes of the cells. So if you talk about the prokaryotic cells alone, they are of different shapes. Cells greatly differ in size, shape and activity. So your mycoplasma, it is a prokaryote, and if you look at the mycoplasma, it has no cell wall. Though it is a prokaryote, so bacteria has a cell wall, right? Gram positive, gram negative have a cell wall. But your mycoplasma has no cell wall, but look at the shape of the cell. So this is your human RBC, so look at the shape of the cell. So depending upon their activity, the cell takes its form, and this is the structure of your neuron. So how do they look? They look pretty different, right? So depending on their function, they are created like this. Why do they have axon? Because they have to conduct and transmit impulses, right? So that is the reason they are designed like this. So your largest cell is your ostrich egg, so that is a single cell. So the neurons are the longest. Neurons are the longest cell, and your ostrich egg is the largest cell. Okay, Satyendra, fine. So moving further, shapes of bacterial cell. You have four basic shapes of bacteria. The first one is bacillus. So bacillus is rod shaped. Next you have coccus, that is spherical, so meaning round shape. So this is rod shaped, this is round shaped. Then vibrio is comma shaped. So how do you write a comma? So it is like this. So this is comma shaped. Then spirillum, so this is spiral. So this is spiral. Okay, so you have your bacillus, which is rod shaped, coccus, which is spherical, and so coccobacillus, this is spherical with your rod shaped combined. So coccus is round shaped and vibrio is comma shaped. So you have spirillum. Okay, spirote is a version of your spirillum, and pleomorphic, meaning it can take any shape. It has no definite shape, but it can take up any particular shape. So that is pleomorphic. So pleo is any, morphic is morphology. It can take up any type of cell shape. So that is your pleomorphic. Okay, so moving forward, so talking about a prokaryotic cell. So these are your bacteria, these are your cyanobacteria. So that is your mycoplasma. Yes, it has lipoproteins. So these include these all come under your prokaryotic cell. So moving further, so all prokaryotes except mycoplasma, they have a cell wall. So this is the first point you should remember. So all prokaryotes, any bacteria, cyanobacteria, so all unicellular organisms have a cell wall, with the only exception being your mycoplasma. So no cell well-defined nucleus. The nucleus is not surrounded by a boundary. Okay, so that is the problem. So no nuclear membrane. Yes, the nucleus is not well organized. The genetic machinery is less complex in your prokaryotes. So it has no well defined nucleus, meaning it has no nuclear membrane. They have extra chromosomal DNA. So that is known as your plasmids. So extra chromosomal DNA is known as your plasmids. So what is the function of these plasmids? So we'll talk about it later, but remember all prokaryotes have plasmids which can be used as a vector. So we are in the era of gene therapy, genetic revolution, etc. If I want to take one gene from me and put it inside Shrimpy, I will not take like this and put it. It is at genetic level. So I need someone to carry it and put it inside Shrimpy. So I use a vector, that is a plasmid. For example, if you want to go from here to Mumbai, I take a flight, I take a train. So similarly, if a gene has to be transferred from my body to Shrimpy's body, then I use a plasmid. Okay. So plasmids are extra chromosomal DNA. So they are extra, they are not the DNA that is needed for the bodily functions or day-to-day activities, but they are extra chromosomal DNA. So no membrane organelles except ribosomes are present inside your prokaryotic cell. Okay. So differentiated form of cell membrane, you have mesosomes. So your plasma membrane is there, yes. So you have your cell membrane and then you have your cell membrane, then you have your plasma membrane. So you have extended foldings of your plasma membrane, so which are called as mesosomes. So imagine in your prokaryotes you have no mitochondria. So mitochondria is a membrane bound organelle, but here you have no mitochondria. So mitochondria is the powerhouse of cell. Then how does a prokaryotic cell produce energy? So it is believed that mesosomes contain some of the enzymes for respiration. So in yesterday's lecture of respiration in plants, I taught you, right? So how respiration yields energy. So much in the same way, in case of your prokaryotic cell, your mesosomes contain those enzymes. So this is a typical prokaryotic cell. Okay. So moving forward. So cell envelope and its modification. So we are talking about the first layer of the prokaryote, that is your cell envelope. Yes, cell wall is there. So now let's talk about the cell envelope. So you have okay. So cell envelope is basically your cell wall. The modifications are you have your glycocalyx, then you have your cell wall, then you have your plasma membrane. It is arranged in this form. Just like in the anatomy of roots, we spoke about epidermis, cortex, endodermis. So it is glycocalyx, cell wall and plasma membrane. So this is how it is arranged. So your glycocalyx is the outermost. So your plasma membrane is the innermost. Okay. So your glycocalyx can be of two types. Okay. So yours based on the cell envelope, your prokaryotic bacteria can be of two types. So it can be gram-positive bacteria and it can be gram-negative. So hold on, I will tell you why they are positive and why these are negative. Just hold on. So glycocalyx differs in composition. So yes, this is in prokaryotes. Now I am only talking about your prokaryotic cell. So after I describe the prokaryotic cell in detail, then we move on to eukaryotic cell. Okay, Anthony Abhi? Yes. So glycocalyx can differ in composition. It can have two forms. So when it is in a loose sheath, it is known as a slime layer. When it is thick and tough, it is known as your calyx. Yes, it is the outer layer. It is outer to your cell wall. So this is your cell wall. So outer to your cell wall, you have your glycocalyx. So inner to that, you have your plasma membrane, cytoplasm and your nucleus. Okay, Shimpi? Yes. So your plasma membrane is selectively permeable. Your cell wall determines the shape of the cell. So this is your glycocalyx. When it is present as a loose sheath, it is called as a slime layer. When it is present as a tough and thick layer, it is known as a capsule. So what is the order? Glycocalyx, cell wall, plasma membrane. So glycocalyx can take up two forms, that is your slime layer and your capsule. Okay, then your cell wall provides shape and rigidity to the cell. Your plasma membrane, it is selectively permeable. Clear? Okay. So based on the cell wall, you have two types of bacteria. So we call them as gram-positive bacteria and gram-negative bacteria. So what is the major difference between these two? So gram-positive bacteria have a thick peptidoglycan layer. So it is a polysaccharide. So just like you have cellulose, so this bacteria have a thick layer of peptidoglycan, and inner to that it has a plasma membrane. So plasma membrane has a lipid bilayer. Yes, this is the polar head and this is the non-polar tail. Right, so it has two layers. So plasma membrane is the same, but it has a thick peptidoglycan layer in case of gram-positive bacteria. Okay, so in case of gram-negative bacteria, your peptidoglycan layer is in between your plasma membrane. So you have two layers of plasma membrane and your peptidoglycan is sandwiched between the two. So this is the major difference between gram-positive and gram-negative. So why is it called so? Hi Munna, yes, hello. So thank you so much for joining my session. So please do like, share and subscribe. Yes, I am very good. How are you? So please stay safe. I heard it's raining in your area. So please stay safe, okay? But why it is called as gram positive and gram negative? So a person called Hans Gram discovered this. So that is why the first term gram. But why positive, why negative? So this is a bacterial cell wall, right? So if I give a particular dye to the cell, yes, so the dye penetrates inside the cell. Okay, but once I wash, the dye does not come out. So imagine I am putting mehendi in my hand. So after 1 hour, 2 hours, I wash my hands, but the stain stays back, right? Yes, the outer layer of mehendi, the black or the brown one goes off, but the orange color or the red color sticks to my palm. So much in a similar way, when I am inserting a dye inside the cell, the dye penetrates through the peptidoglycan layer and it goes inside the cell. Now when I wash the cell, the dye does not go away. It stays inside the cell. So that is the reason it is called as positive. In a gram-negative bacteria, when I give a dye and when I wash the cell, the dye washes off. The dye moves away with water. This cell is not able to retain the dye because of a thin peptidoglycan. So that is why it is called as a gram-negative bacteria. Is this clear? What is the difference between gram-positive and gram-negative bacteria? Did you understand? So this is used for your diagnostic purposes. So based on the dye, based on the retention of the dye, they will check if your infection is caused by gram-positive bacteria or gram-negative bacteria. Hydrophilic, hydrophobic. So this is your hydrophobic because it has your non-polar tail, and this is hydrophilic. Okay? Yes? Clear? So why it is called as gram-positive? Why it is called as gram-negative is clear? No doubts? Yes, good. So you have mesosomes. So I told you they are infoldings of your plasma membrane. Yes, you have a plasma membrane. So they are infoldings of your plasma membrane. They take up the function of your mitochondria. They are localized infoldings of your plasma membrane. So they perform the function of your mitochondria. They are believed to have some enzymes for respiration. So they take part in respiration. They also play a role in reproduction. How? We will talk when I am discussing about reproduction. So these mesosomes are foldings, right? So whenever there is foldings inside your body, that is to increase the surface. So your villi has your microfoldings, right? So this is to increase the surface area of absorption. So similarly, mesosomes, which are infoldings of your plasma membrane, so this also increases the surface area. So now talking about flagella, pili and your fimbriae. So these three sound similar, but they are not. So bacteria can be motile or non-motile. Your bacteria can move, otherwise it cannot move. Okay. So if they are motile, if they can move from your stomach to your intestine, then they should have some locomotory organs, right? So if I want to walk from here to there, I am having my legs, so I am walking. So similarly, for a bacteria to move from my stomach to my intestine, it should have locomotory organ. So a bacteria is considered motile if it has your locomotory organ, that is flagellum. So this long chain-like or thread-like structure is your flagellum. Your flagella has three parts. It has a filament, hook and a basal body. So what is pili? So pili are elongated tubular structures. They are made up of special proteins and they help in reproduction. Okay, so these are pili. Okay, so fimbriae, they are small bristle-like fibers. So these small fibers are fimbriae. They are made up of filamentous protein. They help in the attachment. If my bacteria has to cause infection, it attaches to my epithelial surface. For that attachment, it uses the fimbriae. They are small bristle-like fibers sprouting outside the cell. Clear? Yes. So the next one is your ribosomes. So ribosomes are present in the plasma membrane of the cell because there are no other compartments in your prokaryotic cell. So your ribosome, it has two subunits. It has a bigger subunit and it has a smaller subunit. So the bigger subunit is your 50S and the smaller subunit is 30S. So your flagella is used for movement, your fimbriae is used for attachment. So pili to some extent is also used for attachment. Of course, bacteria is living, that is why it is causing infection, it is taking all our nutrients. Okay, Shimpi? So ribosomes have two parts, larger subunit, smaller subunit. So larger subunit is 50S, smaller subunit is 30S. So what is S? So S is sedimentation coefficient or Schwarzschild unit. So what does this mean? So when you centrifuge this at a high speed, so depending on the density, you get this value. So this is 50S and this is 30S, and when they join together for translation, they together become 70S. So this talks about the density of the ribosomes. Okay, so they join together with the help of a cofactor, that is magnesium. Done? So ribosomes attach to a single RNA. So if you have an RNA, so ribosome goes and attaches to the RNA. So thus it forms a polyribosome. So poly means many. So this is how it attaches. So this is your RNA. So it attaches to the RNA. So ribosomes are important for protein translation. Is this clear? Yes. So with this I have completed some portion of your prokaryotic cell. So some more is remaining, so which we will continue on Monday. So with this I will stop. So please read your NCERT, that is very important. So meanwhile, don't forget to join me for tomorrow's lecture. Okay, inclusion bodies I will teach in the next class. So tomorrow this is my schedule. So 4 p.m. we are talking about glycolysis, and 6:30 we are having a mega mentee, that is for your syllabus wrap up series, and the winner of this series will get access to a mock test papers. Okay. So tomorrow the test is on morphology, anatomy, animal tissues, that is your epithelial, connective, your muscular and neural, and of course cockroach at the end. So with this we come to end of today's lecture. So if you genuinely liked my way of teaching, so please do like, share and subscribe, and thank you so much for joining me at this late hour in the evening. So please stay safe. So take care, and I'm waiting to meet you tomorrow. So this is me signing off. So until then, bye and take care.